the genes that are responsible for G1 cycling and G1 cyclin-dependent kinases, which we talked about the last case. So that would be important because the cyclins are always present, and of course the cyclin-dependent kinases fluctuate and go back and forth. So when you have two of those being affected by the variety of growth factors that we have in our body, you can imagine what would happen in the cells. Okay? So, and like I said, some growth factors, as in the example of G1-CDK, are inhibitory. Okay? So some of these are just examples here that we're looking at. So this is just a regular kind of diagram for what happens when we have receptor-mediated growth factor signaling. And so once again, you have your ligand attaching to your receptor, which is the tyrosine kinase, obviously, because of the structure here. And so this is going to be probably designated as a G-protein because we have that GDP present there. And so when they come together down here, what you have is you have RAS or some other, I guess I'll call it an oncogene, but you don't even have to know that yet. And so then you get conversion here and you get downstream signaling and other things that are happening. So it goes from the membrane here to this. And then you get a phosphorylation cascade. And so what ends up happening at the end of this is that within the nucleus, you get the activation of transcription, okay, and the activation of early genes in essence. And some of these factors include cyclin-dependent kinases and cyclin and elongation factors as well. So if this receptor-mediated signaling here has the ability to eventually affect these, then that would explain why these growth proteins are in cancer and regular cell cycle. So growth factors, and this is just another example. Okay, so what we're looking at here, and we've gone through most of these. This is just a different receptor kinase in that it's a serine reading specific protein. And so they call this one protein kinase B or PKB, and the acronym or the other term for that is AKT. Okay? So these are used interchangeably. And so if we look at the growth factor in this case, what we have is we have sort of a little bit of a different setup for serine reading specific kinases. But nonetheless, we have all this phosphorylation that needs to take place in order for P1-3 kinase to be activated, okay? And so P10 is basically a tumor suppressor gene. And so that's indicated in prostate cancer. And so basically when this P10 can't operate because it keeps cells from going out of control, when that's affected, what you get is, so here, it's working properly, okay? And so what happens is these arrows within any diagram, if they go this way, it means activation. If they have that straight line at the end, it means they inhibit. So in this case, what happens is you get apoptosis being inhibited, and you get cell cycle rescue. So we don't want that to happen within the system. Okay? So regularly this would work to increase apoptosis and to increase cell cycle. So this is your summary of the regulation cycle. And although this looks a little bit complicated, just focus on the parts in here. We know that all this is interphase, and then we have mitosis. We mentioned that the anaphase promoting complex was important for a couple of reasons, and that was for that transition from M to G1, and because of its degradation properties within the mitotic cycle. Okay? So in review, we can say that cells grow basically until they reach their full size, and they stay at that size in G1, or
by mistake, yeah. Exactly. Thank you. And what is the channel setting? 101. Okay, thanks. Sorry about the interruption. And now I can speak much lower. Okay, so cyclin, which activates cyclin-dependent kinases, also phosphorylates retinoblastoma, which allows S-phase proteins to be made. Okay, so there's all these activities in the cell, which basically we want to keep that cell cycling at some points, but we also want to use these checkpoints to keep things in check. Okay, and so we also must remember that there is a supply. There should be a supply of nutrients, and that's very obvious in our own bodies, and also very obvious in a lab setting when you're doing cell growth, because these cells will die without proper food, nutrients, or being fed every couple of days, okay? And also, the other thing that's important in the cell cycle is DNA damage, okay? So, for the example of P53, it inhibits cyclin CDK, so S-phase proteins are not made, okay? So that's another form of cells staying in where they should be. And so there are also checkpoints at G2 and M. And so the one at G2 is mostly to check for DNA damage, which occurs right about here. And the in the M phase, of course, there's all sorts of things happen, but mostly they check that the sister chromatids are properly divided. So if there is any defect in that division, basically what happens is that you get degradation by proteases or other enzymes, and you get that cell either being eliminated by apoptosis or degraded in some form, okay? But hopefully you would not want that cell to continue on if there was DNA damage, and as we do more about cancer cells, you'll see that cancer cells are not really selective. They don't care if there's DNA damage, and so that's what causes the problems, is they grow and produce while you have damage in these cells. Okay. So now we're going to take that knowledge that we have of proteins, receptors, cells, and other things, and see how we can apply it to and understand cancer a little bit better, okay? So cancer is basically the uncontrolled cell proliferation that occurs in the body, and basically things just get a little out of control. And so basically what you have is a normal growth, and this is very basic, but you get shedding of dead cells, okay? And with tumor growth, of course, this does not happen. So what happens is you get the formation of a tumor. And so how does this cancer spread? So basically, if there's no control of proliferation or cell growth, cancers invade the surrounding tissues and also the blood vessels. So they're and cancers are really sneaky too, because they can create their own supply of food and blood. And in order to survive, they have to have that. So they will create their own vascular systems, okay? And so for those that have the ability, and they have different properties, different cells have different properties, for those that have the ability to break off, and this has to do with adhesion versus whether or not they can float around, basically they break off and they can spread throughout the blood vessels. And if you think about the flow of blood in the body, you can imagine that if you have cancer cells floating around in your body, and you're in an immunocompromised state, and you can't fight off that, occasional cancer cell, then they lodge because, once again, of their properties, so they lodge in different areas, and this is where metastases occurs, or that second phase of cancer. Okay, so there's always a primary site, or there may be multiple sites, but usually it starts in one area and then grows and spreads, just like we see here in this diagram. Okay, so cancers are classified in general in different ways. We know that they can originate in any organ, but carcinomas basically arise from epithelial cells. And so examples of this would be lung, breast, and colon cancers. And sarcomas are more of what we hear of osteosarcomas and things like that. And they arise from supporting cells, which would include fat, bone, muscles, things like that. And then we have lymphomas and leukemias, which arise from blood and lymph cells. Okay, so that's just a basic overview, and probably good to know these terms as you go forward, because you hear people being diagnosed of all different sorts of cancers, and usually you can get a feel for what it is, but you know, some people say osteosarcoma or lymphoma, leukemia. Now you know that they're termed based on where they originate. And cancers are caused by several factors, okay? And so basically cancer is complicated because it's a multifaceted disease. It's not only one disease, it's caused by many things, and it's also, sometimes it needs a double hit, what they have a double hit hypothesis. So not only do you need a defect in one gene, you may need a defect in probably 2 genes, and it may need to be combined with an environmental factor in order for that to progress to the state where you get disease, okay? So some of us, or a few of us, like they said in the movie, if we live long enough, we'll get defects that will cause cancer, but it's the ability to correct these defects that will save most of us and keep us healthy. Or if we get 2 or 3 things happening at once, then you get cancer developing because of that load that is on the body. Okay? So, needless to say, they have a really hard time saying that certain things cause cancer. But the one thing they know for sure is that smoking does, and they can't really, I guess, say it's a cause, but they can say it's an association, okay? So that's why you'll never hear definitive statements other than the one by smoke and inhalation on cancer. But they do know that radiation, of course, is involved in environmental toxicants, things like that, okay? And we also know now that mutations in genes can be inherited. And that's where you get that inherited form of cancer. So I'll explain this a little bit later, but also what I want you to understand is that all cancers are genetic. And I don't want you to be confused by that statement, because what they mean when they say that is that they have a genetic origin, which is a defect in the DNA. But inherited cancers are much different. So while 100% of cancers are genetic, only 5 to 10% are actually inherited genetic defects, okay? So the other thing that's important is viruses. And so we now know that human papillomavirus, for example, is a cause of cervical cancer. And that is basically because that virus basically lodges itself in a manner that affects DNA. And so most of you in here are probably from a vintage or an age where you've been vaccinated. And so that's the kind of advances that we make in science as we learn these sorts of things, okay? So I want to focus for a minute on the term oncogene. And has anybody heard that term before or become familiar with it in your courses, okay? So that gets a little bit confusing because they also talk about proto-oncogenes and other things. So we're going to review a little bit about cancer genes. And so basically a proto-oncogene is a normal gene that can become an oncogene due to mutations or increased expression, okay? And then an oncogene is basically a gene that has the potential to cause cancer. And so of the 25,000 genes that we have in our body, I think they've classified about 300 of those as being oncogenes. And trust me, there's a lot more to come. It's only because they've gotten this far in the research. So in tumor cells, oncogenes are often mutated or expressed at high levels, oncogenes are activated, they can cause cells that are usually designated for apoptosis to survive and proliferate, okay? So basically what they do is they change all those cellular pathways that we've studied in normal cells, and they basically create havoc for the cell, which causes it to think that, okay, I must survive at this point, I must proliferate, but basically it ends up to be a non-controlled growth of cells, okay? Tumor suppressor genes, on the other hand, which we've learned a little bit about, they are protective in their role and basically prevent the cell from making that step into being defective or turning into cancer, okay? So basically they function to keep cells in check and to basically let cells go into apoptosis. And so when you get a loss of function, the cell can progress to cancer. Now, tumor suppressor genes usually need something else to happen in order for them to be turned off, I guess. So these are fairly important, okay? And so the loss of these genes may be even more important than proto-oncogenes, because think about it this way. These ones can be turned on, but if this guy's working really well, he can actually take over that concept of dysfunction, okay? But if both of them are not working, then you have real problems, okay? So I mentioned that all cancers can develop because something has gone wrong with one or more genes in the cell. And that's so true. So basically what we're saying about this is that all cancers are based on genetics. But mostly what we're talking about there is genes in general. We're not talking about inherited genes. Only about 5 to 10% are due to inherited cancers, and inherited cancers are basically the result of germline mutations, or something that's passed down from parents or grandparents or any of your lineage, okay? Sorry. So this is just a diagram about how oncogenes measure or trigger cancer, usually by making faulty growth signaling proteins, okay? So we have to keep in mind that these genes eventually want to make proteins, and these proteins, which they called oncoproteins, can also cause defects in the cell cycle. So we know that genes produce RNA and proteins, and we also know that these can create defects in DNA, which can affect transcription and translation. But any of these defects in any of these along the way could cause something to be dysfunctional, okay? And that could include regulatory transcription factors, steroid hormones, CREB signaling pathways, or any of these control elements, for example. And it could affect the peptides, the proteins, transcription factors, histones, the end proteins, cyclins, growth factors, G proteins, etc. And so basically, if you have these mutated genes, in the end, what they do is they produce faulty proteins. And faulty proteins, of course, if they're not eliminated from the cell, and that's another cause too, because those things that eliminate proteins are produced as well. So you have an accumulation of defects that can eventually cause dysfunction, and in most cases lead to disease, but because we're talking about oncogenes, the disease that we're referring to, of course, is cancer. So some of these oncogenes are grouped together by protein function. So think of all the things that genes do. And so basically what they do is they produce growth factors, receptors, they produce plasma membrane G proteins, non-receptor protein kinases, transcription factors, and they also produce cell cycle or death cell regulators, okay? So just a note here that they are grouped by protein function, and these are some of the most common ones, or some of the ones that we talk about a lot. And these are the common cancer types that are produced from these, okay? So I want to take you back to the definition of oncogenes and tumor suppressor genes. And so what I want you to think about is that when we think of normal genes and oncogenes, basically what you want to think about is consider them like a car, and consider oncogenes like the gas pedal, okay? And so basically in normal function, you have full control of your car. You can put down the gas, you can step on it, you can take your foot off it. It controls the rate at which your car moves, okay? But when oncogenes are activated, basically your gas pedal is stuck. You have no control over your car, and you're going at fast speed, and basically what you're producing is the ability for you to run into somebody or to, basically produce a bad situation. And for oncogenes, that bad situation is basically cancer, okay? And I've outlined some of the important oncogenes here, like HER2/neu. Most people will hear about that if they study breast cancer, RAS gene, MYC, SERP, which was the first oncogene ever discovered, and they did that with work on chickens, because they basically found out that the SERP oncogene is present, but becomes activated, and then chickens, go into a cancer state. And the other one is an enzyme that codes for telomerase, which is called hTERT, okay? So, and then for the tumor suppressor genes, what you have to think about is compare these to the brakes on your car, okay? So basically, in a normal function, what you have is you have the ability to slow down your car, okay? And so these tumor suppressor genes have the ability to produce things, but when they are not active, basically they have no brakes and they can't control, basically there's a loss of that ability to regulate cells, and so what you get is you get an active oncogene being involved in this. You get no brakes on the car, and eventually what you have is uncontrolled cell growth. And some of the most important ones for this are P53, which regulates cell division and cell death, retinoblastoma, which alters the activity of transcription factors, and APC, which controls the availability of the transcription factors. So all of these things function, just like we've seen in proteins, to do different things. And so now we have it that we have these genes, or proteins, or otherwise, that when they are dysfunctional, basically they're causing a disease state. Okay? How much time do we have left? Just a few minutes. Okay. So proto-oncogenes help regulate, here's my typo of the year, cell growth and differentiations. And the resultant proteins are called oncogenes. Okay? And we've also learned that proteins are involved in signal transduction and mitotic signaling. Okay? So it's no different for these. Proto-oncogenes are involved in these processes too. And so once they're activated, they become tumor-producing agent. And they require additional steps. We mentioned this, the two-hit hypothesis. Basically, they sometimes require two steps, such as environmental factors or a viral infection, so that they can go on to produce cancer. Okay? So when we talk about risk and how to eliminate risk, that's why we control some of the things that we can control inside our body. So you can't really control what you inherit. You can't really control some of these DNA problems that we have or defects. But what you can control is some of the environmental factors surrounding these. Okay? And this is just a summary of some of these oncogenes once again, and how they're related to breast cancer, esophageal, colon cancer, pancreatic cancer, liver, and melanomas. Okay? So this is just another example of how we know that genes are needed for S phase. Genes needed for S phase are not transcribed, and so then basically you get that restriction point defect where you can't get from G1 to S, or you get from G1 to S more often than you want to, okay? And these are just examples that occur within the RAS pathway with the use of a phosphorylated ERBB protein, okay? And this is just another example that cancers behave in such a way that they can divide in the absence of growth factors. Okay? So all these things that are normally in the cell to control and kind of put a stop and go system within place, cancer cells somehow have properties that can overcome this. So they don't really care if there's growth factors in place. They can overcome that need to divide. They're not responsive to internal conditions such as DNA damage. So normally you have normal cells, which basically you can say, oh, there's something wrong. Cancer is basically saying there's something wrong, but I'm still going to produce this. And cancer cells can block apoptosis. So that's one of the main things, is that cells keep proliferating and going on. And so I think we have 2 more slides left in here, and I can do those the next stage, but I know that you're all anxious to get to the final exam piece, so we'll start this right now and we'll finish it on Friday. And so basically you can treat your last section, which we've done, which is lectures 26 to 32, and tutorial 5 as an interim number 3. Okay, but it's going to be in the form of a final exam. And so for that section, you'll have 25 multiple choice, 15 fill-in-the-blanks, and 10 matching. And the only matching that I can pick for this are gene and gene category, functions of enzyme, and features of protein synthesis. Okay, so that gives you a good idea of what those matching will be about. And there will be a bonus on this section as well. And so for all other sections, which are only 2, we'll combine, we'll have 25 multiple choice for those, 15 fill-in-the-blank, and 10 matching. We know that we're going to have about 5 on glyconeogenesis, which is a complete giveaway, and the enzymes involved. And I'm also going to put a really easy one in there on cell structure and function. Okay? So it might be nucleus, what does it do? You know, chloroplast, what does it do? Lysosome, what does it do? So make sure that you know your eukaryotic cell and the function of each. And so the exam will be worth 100 in total, plus the 2 bonus points. And I just wanted to put up this cartoon because I find it really amusing that most of you study about, 80% or 90% of what's in the course, and then this cartoon depicts things that are on the exam, and we all are afraid to fall into that trap. But if you read your notes from this class, which are your lecture slides, and review your tutorials, this won't happen, okay? So on Friday, and I apologize for having to do the class evaluation today, but I'm just afraid of the weather on Friday, and we have to get that in. So we'll do a review on Friday, some really good tips for the exam, and we'll go from there.